when he was involved in studying some of the molecular pathways associated to AD pathophysiology using lymphoblasts from patients. In the year 2010, he moved to the UCL Institute of Neurology in London, where he analyzed mitochondrial bioenergetics and dysfunction in cells from frontotemporal dementia and ALS patients, under the supervision of Drs. John Hardy, Andrei Abramov, and Hélène Ploufavreau. In 2014, he came back to Spain, where he joined Eva Carro's group at the 12 de Octubre and CIBERNED, and he's been interested in studying the alterations in molecular pathways and in the search of biomarkers for neurodegenerative diseases, including AD, PD, FTD, and ALS. Since last year, he's Miguel Servet Fellow at the 12 de Octubre Research Institute, and he will talk today about mitochondrial dynamics and autophagy in AD models. So I would like to thank Fernando for his willingness to share with us his work, and welcome to the CIBERNED Webinar Series. Fernando, the floor is yours. You can start sharing your screen, and please go ahead. Thank you. Fernando, excuse me, you have your microphone off.
I'm sorry.
All right.
Thanks, Miguel, for your presentation, and thanks, CIBERNED, giving me the opportunity to show my part of the work we have carried out in Eva Carro's group. So as Miguel has mentioned, I'm gonna give my talk about mitochondria and autophagy that we have showing some experiments that we have done using several Alzheimer's disease models. Sorry, I don't know why the presentation is not working. Here we go. Okay. As most of you know, can you hear me? Okay. As most of you know, the most accepted hypothesis in Alzheimer's disease is the amyloid cascade, which establishes that the A-beta peptide could be the cause of the disease. But it's not, what is known is that A-beta accumulation occurs earlier or years before the onset of the clinical symptoms of the disease, but it's not clear if this accumulation is the cause of or the consequence of AD pathology. What is clear is that A-beta accumulation exerts toxic effects on the brain cells. This A-beta accumulation may be removed using an autophagy-dependent mechanism that indeed remove the excessive production of A-beta peptide. But also some studies indicate that A-beta peptide accumulation can be an autophagy trigger itself. In addition, there are also a number of studies that indicate that A-beta could be, in addition of autophagy, may induce the autophagy-specific clearance of mitochondria, which is known as mitophagy. Mitophagy, as I just say, is the specific autophagy for the mitochondria. When a damaged mitochondria is accumulated in the cell, they initiate a protocol of elimination or clearance that is known as mitophagy. This damaged mitochondria can occur because alterations in the mitochondrial physiology, and the alterations in the mitochondrial physiology in Alzheimer's are already known. But what is not known is if they are an early event in the disease progression, or also if they are associated to amyloidosis or other possible conditions. Mitochondria are very important in cells, and particularly in neurons, because as they control numerous cellular processes that includes the control of calcium homeostasis, the ATP production, and cell death. And in addition, the accumulation of dysfunctional mitochondria could be very toxic for cells. So we decided to study the effects of A-beta peptide accumulation on the mitochondria in several Alzheimer's disease models, starting by the known animal model APP-PS1 mice. We checked the effect of A-beta in the mitochondria in the common affected areas in Alzheimer's disease, or in this, which are the hippocampus and cerebral cortex in mice from 3, 6, 12, and 12-month old, and to see the progression of these possible effects. This APP-PS1 mice is known that has cognitive and memory impairments detected before 6 months old, and these mice show accumulation of A-beta toxic peptides. For example, I saw here the accumulation of A-beta 40 and A-beta 42 in the cortex and the hippocampus in our mice, that is detected from 3 months and is already more significant at 12 months old. So to study how the status of the mitochondria in these tissues, what we have done is a general picture about the amount of mitochondria in these animal models. So to have this picture of the amount of the mitochondria, we have used the mitochondrial structural protein, the complex, this beta subunit of complex 5, to see the levels of the mitochondria in the cortex and the hippocampus of these mice. And we found that the amount of the mitochondria in these animal models was starts to be significantly reduced in the hippocampus at 3 months old, in the APP-PS1 mice compared with the wild type, and at the 6 month old animals in the cortex. This reduction of the mitochondria occurs also in the wild type mice, but so this could be an age-dependent effect, but this occurs much more later than in the APP-PS1 mice. We can conclude that this is an effect of the amyloidosis, because when we treat different cell cultures with the oligomeric A-beta 42 peptide for 24 hours, we found that the levels of this structural mitochondrial structural protein complex 5 beta subunit were reduced in both in primary neurons and in the SHSY5Y cells. This the amount of the mitochondria, or the mitochondrial mass in cells, is controlled by a process which which is known as mitochondrial dynamics. And this process includes the mitochondrial biogenesis and the mitochondrial fusion and fission. These two processes in healthy cells are in a balance, but when this balance of mitochondrial biogenesis and mitochondrial fusion and fission is not equilibrated, may trigger neurodegeneration. But also particularly the mitochondrial fusion and fission process is in a equilibrium in the cells, and when the mitochondria or one mitochondria which are forming a network is damaged, this mitochondria is split from the rest of the network and starts a program that send this mitochondria to be eliminated by mitophagy. So that's fine, but when mitophagy, the process of mitophagy does not work properly, it may result in accumulation of damaged mitochondria that also may cause neurodegeneration. So we check if this reduction going back to our model, we check if this reduction in the mitochondrial mass in the APPPS1 mice was related to changes in the mitochondrial biogenesis or the mitochondrial fusion and fission. And we first analyze this in these processes in our mouse model. We check first the mitochondrial biogenesis, analyzing the levels of this master regulator of mitochondrial biogenesis, PGC1 alpha, and we found only in the brain cortex of APPPS1 mice, we found that a reduction in this protein from the 3-month-old mice, and this reduction was maintained during the age of these animals. Such reduction did not occur in the wild-type mice, and we did not find changes in the hippocampus. So the other process, the mitochondrial fusion and fission, was checked by analyzing several of the mitochondrial fusion and fission controlling proteins. For example, I saw here that the levels of the fusion proteins, mitofusin 1 and mitofusin 2, were significantly decreased or reduced in the APPPS1 mice, especially in the cortex, from at early ages, at 3-month-old, compared with the wild type. And this reduction was maintained during the age. And on the contrary, the fission protein, check the fission protein DRP1, we found that at least in the cortex, the levels of this protein were significantly increased at 6-month-old. So summarizing, we can say that reduced fusion protein levels that are shown here, mitofusins 1 and 2, are accompanied by increased fission protein levels, and both the balance of these changes in the levels of these proteins may trigger mitophagy. So what we did next was to check the mitophagy in using the SHSY5Y cell model, and we added to these cells the oligomeric A-beta 42. We did some immunocytochemistry experiments, and what we checked were the levels of these autophagy common markers, P62 and LC32, and only we quantified the colocalization of these two autophagy markers with the mitochondria. This is actually what is shown in the bottom part of the screen. So we found that both autophagy markers colocalizing with the mitochondria were increased under the incubation with A beta, and then we added the blocker of autophagosome lysosome fusion. We use bafilomycin to see whether we could find an accumulation of these autophagic vesicles. And indeed, we found that such accumulation occurs in the untreated cells, but also was much more significant in A beta. To see whether this was an effect of autophagy or was also indeed an effect of mitophagy, what we found is that checking the levels of the mitochondrial structural protein complex 5, we and consistent with the previous results in this immunofluorescence, we quantify the complex 5 levels under incubation with A beta, and we found that they were reduced. But then the use of bafilomycin showed that so an accumulation of this protein, indicating accumulation of the mitochondria. So what we can conclude here is that A beta overload triggered mitophagy. And because I mentioned before, this A beta is known that also is an inductor of autophagy. We check in our cellular models that the effect of A beta in general autophagy, and as you can see, so in the both in the autophagy indicators P62 and LC32, we found that A beta increased the levels of these two markers, and then with bafilomycin we obtained the highest accumulation of autophagosomes. We found this effect in both in SHSY5Y cells and also in rat primary neurons. We finally checked the autophagy in the animal model, and with the exception of P62 in the cortex, we found that both markers, P62 and LC32, were found increased from the six-month-old animals, significantly increased compared with the wild type. Here you have the P62 levels, and here the LC32. So in conclusion of this part, we can say that A beta peptide produce or exerts a dual effect in as we have shown in vitro and also in our in vivo models, and the mitochondrial dynamics imbalances we found in the models end up increasing mitophagy. And this is this effect, dual effect, could be extrapolated to in vivo, as you can see, and is we can say that is an early event in the pathology. So I would like to note that also at older ages, despite increasing autophagy, greater accumulation of A beta peptide is observed, suggesting that the production of A beta may exceed the elimination levels. I want to, so this is these experiments, you can find details of this work in the recently accepted work in scientific reports that will come out in the next days. Also, because the title of my talk is related to mitochondrial dynamics and autophagy in Alzheimer's disease models. I want to show you some other preliminary data we have in the group that are related to other that are done have been done with other cells from Alzheimer's disease patients. We have used fibroblasts from several Alzheimer's disease patients that particularly they are SOL1 mutations carriers. So it is SOL1 is a protein, or is a SOL1 gene encodes this protein, this sortilin related receptor that is highly expressed in the brain. And SOL1 mutations is known that cause autosomal dominant early onset Alzheimer's disease, but also SOL1 variants is known that increase the risk of late onset Alzheimer's disease. In addition, low levels of this protein have been found in sporadic cases of Alzheimer's disease. This protein, SOL1, binds the APP protein, and this interaction is prone to non-amyloidogenic pathway of APP. In addition, this SOL1 protein, because it has several functional domains, one other domain of SOL1 binds A-beta peptide and contributes to A-beta lysosomal clearance. And so reduced brain levels of SOL1 may alter APP transport and processing, increasing the A-beta peptide generation. Recent work or carried out, I think it came out in 2020, carried out by Rick Livesey group in the UCL in London. They show that this deficiency in SOL1 is related to the deficits in the autophagy degradation mechanism. But also what took our attention was another work carried out by Berthersol, in which they saw that a SOL1 deficiency is related to mitochondrial dysfunction. So putting together these works, we decided to study the autophagy and mitophagy in AD in patients in these patients with Alzheimer's disease carrying these SOL1 mutations. So because in consistent with the previous work, we analyzed the autophagy process in our fibroblasts carrying the mutations, and just checking the P62 autophagy marker, we use several autophagy stimulus and in our cells, and we found that under starvation and by addition of rapamycin with induced autophagy, when we compare the levels of this autophagy marker P62 in our cells, we found that in both cases, the autophagy P62 levels were much more higher than when compared with the cells from healthy donors. Indeed, also in the untreated cells, we still found a higher levels of P62 markers in all Alzheimer's disease patients carrying the SOL1 mutations. So at least our cells are consistent with the previously reported results that this SOL1 deficiency is related to deficiency in or disrupted autophagy. So we next wanted to check the mitochondrial if in agreement with this other work from Bethersol, if these mitochondria were affected, and we check first the mitochondrial membrane potential by analyzing the tumor inflorescence. The mitochondrial membrane potential reflects the mitochondrial health and function, and we found in all patients carrying the SOL1 mutations, the mitochondrial membrane potential was reduced, as you can see also in the quantification of the tumor and the pictures. So when these mitochondria are depolarized, as you see here, the depolarized mitochondria starts the program of mitophagy. So we next check in some of the in these cells from patients the mitochondrial mass. And what we found is that the mitochondrial mass in these patients was significantly, almost in all of them, was significantly increased compared with the average mitochondrial mass found in the cells from healthy subjects. So we think that this could be because damage in the mitophagy process, and indeed when we added to the cells the baphyllomycin, we found that in all control cases, baphyllomycin increased the mitochondrial mass because baphyllomycin blocked the fusion between autophagosomes and lysosomes, but apart of, with the exception of patient 3, in none of the SOL1 carriers, the baphyllomycin significantly increased the mitochondrial accumulation. So we can say that mitochondrial flux was disrupted in these SOL1 mutation carriers. We confirm by this result by checking the mitophagy flux by immunofluorescence using the P62 autophagy marker, and we estimated only the colocalization of P62 autophagy marker with the mitochondrial structural protein beta subunit of complex 5. And we see that this, and we check we show here the ratio between the condition with baphyllomycin and the condition without baphyllomycin. You can see here, that the levels of the autophagy flux was significantly reduced, at least in 3 of the SOL1 mutation carriers compared to the control cells. So just in conclusion of this SOL1 preliminary data, we can say that autophagy is disrupted in fibroblasts carrying SOL1 mutations. Also that fibroblasts from SOL1 mutation carriers, so mitochondrial depolarization, and that the mitochondrial mass is increased in most of these SOL1 mutations carrier, and this is associated with disruption in the mitophagy. So just to finish, I would like to mention that the first part of this work belongs to the Macarena de la Cueva PhD thesis project. The second part, the SOL1 part, belongs to Sara Del Rey master thesis project. And also I want to thank people from the group, Desiree Antequera, and people from the CIBERNED group, Marta González, Víctor Blanco, Alberto Villarejo. Also people that are now here in the hospital, Carolina and Estrella, and of course, I want to thank Eva Carro as head of this CIBERNED group. Thank you all of you for your attention. Thank you.
Thank you very much. Very nice presentation. So our paper is up for discussion. So if anyone is interested in making a question, please either raise your hand using the icons in the application, or just send me a message through the chat stating your name and affiliation, and I will give you the floor to make the question yourself. There is no need for you to write down the question, since I think it's more agile if I allow everyone to do the question directly to the speaker. While we wait for the next, for the first question, let me just remind everyone that CIBERNED Webinar Series are held every other week, usually on Thursdays at 3:30 p.m. The program is available through the CIBERNED webpage. So if you are interested in looking at the next webinars, you can go to the website and check the program until the end of July. In the next few days, we will probably have ready, mostly ready, the program for the second semester, and then we will post it also in the web page. So, any questions? I think we have a raised hand here by Isidro Ferrer. Isidro, please go ahead.
Yes, I'm here. All right. Good afternoon. Well, thank you for this exciting presentation. And I'm very interested in one point. I fully agree on the aspects leading to degeneration of mitochondria related to the presence of beta amyloid. But the second part of your talk, I think it raises new aspects that could be important in the pathogenesis of Alzheimer's disease and other diseases. And this is the effect of certain proteins on the membrane, not only on the mitochondrial membrane, but also on the cytoplasmic membrane, the relationship between the endoplasmic reticulum and mitochondria, the so-called MAM. And I would like to ask you whether do you think that part of the alterations that we see are related to the abnormal signaling in the cell membrane rather than in the presence or on the presence of the products. For example, you have seen or you have presented in the second part that salt affects the relationship of the alterations in the membrane before the appearance of any kind of abnormal protein. So it's enough that the mitochondrial membrane is altered, and probably other structures, as for example, caveolae and other membrane structures, to produce this kind of alterations, in spite of the lack, or despite the lack of beta amyloid. And if we translate this part to the first one, I would like to ask you whether you have seen alterations before the appearance of beta amyloid, because what I think, and I thought that you have presented alterations by the end of the third month in EPP-1. So at this age, the animal already has some kind of beta amyloid. So the first plaques appear at this age. But what happens if you study these animals at the age of 1 month before the appearance of beta amyloid? Is there any change in the mitochondria, in mitophagy and mitofusion or whatever? So the problem is very clear related to beta amyloid. But in addition to the beta amyloid, is there any change before the appearance of beta amyloid? This is my question.
Yeah. Thank you for your question, Isidre. You are right, and this would be an interesting issue to analyze these mitochondrial disbalances in the thinking of on the APPPS1 mice. So I don't know who. Yeah, these balances could be studied at earlier than 3-month-old mice. This would be an interesting point, but also I have to say, I think is consistent with your conclusion. I have to say that these cells we are analyzing from AD patients carrying the SOL1 mutations, we have to say that none of the mutations of these 5 patients are in the A-beta related domains of SOL1. I mean, SOL1 is have several functional domains, and one of them is identified as APP binding domain. So if a mutation occurs in this domain, what is expected is a dysfunction in the A-beta APP processing. And also this protein has another A-beta binding domain, which helps to the clearance of A-beta in the lysosomes. And any of the mutations, none of the mutations we have in our patients are in these domains. So this make to think that the alterations in the mitochondrial physiology we found in our patients may not be related to a beta processing. So that's, I don't know if I address your question.
Yes, yes, of course, and thank you very much. And in this regard, I would like to stress, for example, that patients that have Gerstmann-Strauss-Schenker disease, in which there is, in addition to prionopathy, the inherited prionopathy, there is a tauopathy. When you obtain iPS cells from these patients, then you can see that the production of tau, of abnormal tau, in these iPS cells, well, neurons derived from these iPS cells, the production of tau precedes the appearance of abnormal PRP. So that means that an alteration in the characteristics of the structure of PRP may lead not to the production of prion, but rather on the first production of tau. And the same happens, for example, in patients bearing the ApoE. These iPS cells transforming into neurons then produce beta amyloid and tau. But when you block the process of amyloidogenesis, tau remains produced at the same level. So that means the membrane is very important in this part. And often we interpret that all the lesions are derived from the production of beta amyloid, when, as in your case, for example, you have seen that the main abnormalities is at the cell membrane and at the cell or the mitochondrial membrane. So I'm very, very happy to know your point.
Yeah.
And thank you.
Thank you for you. I want to say that in the next months we are planning to generate direct neurons from this SOL1 mutations carrier by direct conversion of from fibroblasts to neurons using this genome Mertens protocol, and we will be happy to show the data that we obtain with this eye neurons in the next months. Thank you.
Thank you.
All right, thank you both, Isidro and Fernando. So we have another question here from Nadia. So Nadia, you can make your question yourself directly to Fernando. If you state your name and affiliation, please go ahead.
It's
Nadia?
Yeah, hello, can you hear me?
Yes, now
yes.
So thank you so much, Fernando, for the interesting talk. Was interesting about the possible role of the astrocyte. So in this mitochondrial dysfunction, I don't know if you check the level of the mitochondria in the primary astrocyte culture, as you did in the neuron culture, and if you think, if the astrocyte can play a role in this biogenesis and fission phasal alteration in your model or not. Thanks.
Thanks, Nadia, for your question. I have to say that we haven't checked the mitochondrial dysfunction in the isolated culture of astrocytes. We can presume that maybe this the possible mitochondrial dysfunction will go by different ways, because as you say, the astrocytes are not as oxidative phosphorylation dependent as occurs in neurons. So we would expect different phenotypes, but it's something interesting, of course, to analyze.
Thanks.
All right, thank you, Nadia, Fernando. Any other questions? I don't see any questions coming through the chat, nor any raised hand. Okay, no more questions? I can hear someone talking, but I'm not sure whether it's any interest in making a question or just a micro which is on. All right, I don't see any questions, so I think it's time to close the session. No, before once again thanking Fernando for his willingness to share his work with us, and for all of you for attending. So I hope to see at least some of you next week in Salamanca in the meeting. Otherwise, we'll be back here in the next webinar in 2 weeks' time. All right, so thank you all. Thank you especially to Fernando, and have a great day. Bye-bye.
Thank you.
Thank you very much. Bye-bye.
Bye-bye. Bye.